Okay. Good evening, everyone. It's already 7:13. I think I should not wait more. We'll start with today's class. So today we are going to discuss some questions related to week 2. Okay. So yeah, whoever has joined new to the session, welcome. And whoever already has joined in week 1, they know the rules. First rule is that please do not unmute your audio or video throughout the session. I should be the only one speaking throughout the session. If you have any doubts or queries regarding the questions, then you can type your questions in the chat box. Okay? Chemical kinetics are feeling so tough to study. Can we skip? Skipping that is not in my hand. If you wish to, you can skip though. But today I have not kept any chemical kinetics questions as such. So my all my questions are going to be theoretical or very less numerical. We are not going to discuss that much of numerical questions, okay? Because it will take time and you have to solve it and then answer. So I'm avoiding those kind of questions in these sessions, okay? So whatever it is, it will be fact based or theory based, okay? So yeah. So I hope everyone knows the rules. So the first rule was to keep yourself mute throughout the session. Second is I will be showing you the question. You will be given around 3 to 5 minutes, or depending on the level of question, you will be given some time. You have to think what is the correct answer for this question. So all our MCQs, you have 4 options for each question. So you have to think which is the correct answer according to you, and you have to write it down in the chat box. Whatever you think is the correct answer, write it down in the chat box. A, B, C, or D, whatever. No need to write the full option. Okay. So I hope I am clear with this. So we will start. It's already 7:15. We should not delay it more. And I have uploaded the YouTube video for week 1, and the PPT is also uploaded in the Google Drive. You all must have got a Google Excel sheet link through mail to get access to these sources. So if you check them now, everything is uploaded. I have uploaded today only, so maybe if you have checked before, you would not have got it. But now you can see the YouTube video is already uploaded for week 1. So whoever is interested to see the week 1 session again or have missed it, they can go to the YouTube video and see it. Okay. So yeah, we'll start. If there are any questions, you can write down in the chat box. I will start with question number 1. So today we have 15 questions. I thought maybe 20 questions is becoming too much for each session. So I concised it to 15 questions this time. Okay. So yeah, let's start with first question of today. Which of the following is a key feature of a batch reactor? Continuous feeding and removal, plug flow behavior, reactants are charged initially and products removed at the end, catalyst pellets are used inside. Start answering. And whoever is joining, it would be good if you all can join little bit early to the class. Okay, so is everyone done? Okay, so I'm not getting any further answers, so I'm hoping it's done. Let's see what is the correct answer. It is C. In a batch reactor, all reactants are charged at the start and the products are withdrawn only at the end. So C is the correct answer. Here you can see the question is taken from this slide of the PPT. Reactor classification based on mode of operation. So what are batch reactors? Reactants are charged into a vessel at the beginning of the operation and products are discharged at the end of the operation. The chemical reactions takes place over time. And applications of batch reactors are ethanol fermentation, chemical industries including dyes and inks, wastewater treatment, etc. Okay, so I hope this was clear. Moving on to the next question. Which reactor configuration typically shows no axial mixing and only radial mixing? CSTR, PET batch reactor, plug flow reactor, or airlift reactor? Start answering. Excuse me. Okay, I think we have done, we have given enough time for this question. Let's see what is the correct answer to it. It's C. Plug flow reactors, right? So plug flow reactors exhibit ideal flow with no axial mixing, only radial mixing across the tube's diameter. So here you can see this question is taken from this slide called the tubular reactor or the plug flow reactor. So, the reactor consists of a hollow pipe or tube through which the reactant flows, where the reactant moves as a plug along with the pipe, right? So, that is why it is called as the plug flow reactor. And next, what is there? The concentration of reactants varies along the pipe length, right? There is no axial mixing, as the question said, there is only radial mixing. So applications are homogeneous or heterogeneous reactions, product inhibition process, high temperature reactions. Here you can see this is how a tubular reactor or plug flow reactor looks like. So this is the way from where you will, the feed will be entering and from here there will be the product, right? So moving on to the next question. A cascade of CSTRs is often used in industrial applications to reduce turbulence, increase yield and conversion, improve radial mixing, simulate batch operation. Start answering. So as you can see, all the questions are from the slides given for each week. So it is very important that you go through the slides and then come and join the session so that you can answer and understand all the questions correctly. And when we revise the same thing through these questions, you won't get any doubts, because obviously I'm not again explaining the whole concept, because it is already given in the lectures. So it is very important that you attend the lectures and then come back here. Okay. Okay. So let's see. Sorry. Excuse me. Okay. So the correct answer is B. Multiple CSTRs in series can approach plug flow behavior, enhancing overall conversion efficiency. So that's what is the correct answer. Increase yield and conversion, right? So here you can see what is CSTR, that is continuous stirred tank reactor. This is another kind of reactor. Reactants and products are continuously added and withdrawn, while the content within the vessel are vigorously stirred using internal agitation, right? And the cascade of CSTRs can be used for higher conversion of reactants. Applications are biological industries such as SCP, antibiotics, and wastewater treatment. Here you can see how a CSTR looks like. The feed is being continuously added and it is continuously agitated, and then the product is taken out. Moving on to the next question. The fluid in a fluidized bed reactor is used to bubble through a stationary liquid, stir the contents using mechanical agitation, suspend solid particles for better mixing, flow in plug like manner through the reactor. Yes, the live sessions are compulsory to attend. Okay, I'm saying, I said last time also, it is compulsory to attend. Attendance is being taken. I have been reporting the attendance to the NPTEL team. So I don't know whoever is missing what will be done with them. Maybe it has marks or not. I'm not sure about that, but it was told to me that the students have to attend the live sessions. It is compulsory. So the population has severely decreased from week 1 to week 2. So whoever is stopped this, I don't. So there was a feedback form sent to you all regarding these live sessions, right? Last week. So I can't get access to that what feedback you all have given, but I would like to know from you all personally. Is there anything you want more in the session or anything less? You can let me know. If it is possible, I can implicate it. Okay. Are you liking the sessions or is it really boring? Because someone already asked, is it compulsory to attend? Please type your whatever you want to say, type it down. Don't say it, type it down. I'll check. Okay. Abhay Panda. I think you want to say something. You can write down the chat box. That is not allowed. Model question papers, ma'am, or else pattern. That is not my work. Is my work is to make you all revise the week with some multiple choice questions. That was what told to me, so that's what I'm doing. And I only don't have any model question paper, then how can I give you? Or nor I know the pattern. I know that there are going to be MCQs. Actually, I think already the pattern is written in the NPTEL website. I was checking few days back. I think 75 marks of exam is there, and you need to score at least 30. I think something was there. I read. You all can also see that. No, that is not possible. Like, I am not the one to give you notes at all. That you have to make by yourself by at the lecture notes. That thing I can't do. That thing I'm not assigned for. I'm only assigned to discuss questions with you. That's all. I can't give you any kind of short notes. That is not possible. Weekly revision classes we are already doing. The questions we are doing is very important because you're going to get that kind of questions only in the exam. So you can think like that, right? It is equally important to join the revision classes and know these MCQs or practice these kind of MCQs and how you are getting in your assignments as well, right? Because MCQs are going to be a lot in the exam with some subjective questions. Mostly it is going to be MCQs, I think. So, but I can't say you exactly like how many marks you're going to get MCQs and how many marks you're going to get subjective type of questions. But I think you are going to, yeah, I told you, right? I have already uploaded the PPT of week 1 in my Google Drive and you must be having the link for it. I think the NPTEL team mailed you with the Google Drive link last week. If you check that, you can get the YouTube video as well as the PPT. Okay. Okay, so let's see what is the answer to this question. Sorry, it is C. The high velocity fluid suspends fine particles, enabling good mixing and heat transfer. Okay, so so the fluid in a fluidized bed reactor is used to suspend solid particles for better mixing, right? Here you can see the fluidized bed reactor. What is that? A vessel filled with fine particles, that is the size is smaller than 500 mm, that are suspended by the upward flowing fluid at high enough velocities to suspend the solid. And next, what is there? The fluidized bed provides good mixing of the particles and uniform temperature. Typically, velocity 6 to 20 uh, meter per hour. Recycle ratio 5 to 500. Bed expansion is 30 to 100%. So, this is everything you should know about the fluidized bed reactor. So, the question was the fluid in the fluidized bed reactor is used to suspend the solid particles for better mixing, as you can see here, right? So the applications of fluidized bed reactors are multi-phase system with biocatalyst, food processing industries such as peas, shrimp, sliced vegetables. They all used these kind of fluidized bed reactors for food processing. Okay. So moving on to the next question. Which reactor is best suited for aerobic fermentation involving shear sensitive organisms? CSTR, airlift reactor, tubular reactor, trickle bed reactor. Start answering. How both are same? Are we discussing the same set of questions which you got in the assignment? No, right? So here you are going to practice some more questions other than the assignment. That is the motto of this session. That you get to practice some more questions other than your assignment questions. Otherwise, I would have discussed the assignment questions itself. Right? That Abhay Panda, I think I already told that that is not for which I am assigned. If you are really interested in having a one shot theory revision of the week, please contact the NPTEL team that you want like that. If they allow it, maybe they can assign another TA for that, right? So, as I told you, I am already assigned to discuss questions with you so that your topics are revised because I am not discussing question for only one slide. I am discussing questions from almost all the slides present for a particular week. So, through the questions, you are getting revised for the topics taught in the lectures, right? That is the way of making you people revise everything again. Revision doesn't mean again teaching the same thing again, right? That's why it is told that attend the lectures and then come and attend this session. Otherwise, there is no point in joining here if you don't want to go through the lectures only first. If you want me to teach again, then what is the point of having the lectures? Just tell me once, right? That is not correct. That's why I'm not assigned to that kind of a thing. Here, if I discuss questions and obviously I'm giving you explanation, and I'm also showing you the slides from which I have taken the question and we are again going through the theory. That means you're going to a revision class again. Okay? And obviously I have picked up the important topics which will be useful for you all. Right? So I hope it is clear and no one has any doubts regarding changing the pattern of the live session itself. That can't be done. I told you I can implicate only things which is possible, not the impossible ones. Okay, so let's see what is the answer to this question. It is B. Airlift reactors. So these are the reactors which are best suited for aerobic fermentation involving shear sensitive organism. So airlift reactors are low shear systems with high oxygen transfer, ideal for sensitive cells like algae or yeast. Okay. So here you can see what is an airlift reactor. Airlift bioreactors are similar to bubble column reactors but differ by the fact that they contain a draft tube. So we have not discussed what are bubble column reactors. It will come in the next few questions. So the draft tube is always an inner tube or an external tube which improves circulation and oxygen transfer and equalizes shear forces in the reactor. And the applications are photobioreactor for algae cultivation. Like I said, it is used for sensitive cells. Alcoholic fermentation using yeast. So the alcohol, how it is prepared from fermentation through yeast. So for those kind of applications, airlift reactors are used. Moving on to the next question, we are at our sixth question. Which material is not typically used for large scale bioreactor construction? Stainless steel, borosilicate glass, mild steel with epoxy coating, concrete. Already, you all are, I think you all have access to the week 3 lectures as well, right? So, if anyone has gone through the week 3 lectures and they want me to make questions from one particular topic which you think is difficult, then you can let me know that. Then I can make questions on that topic itself which you think is difficult and you need more revision on that topic. That can be done. If you let me know in advance that which topic you want questions on, particularly, I can make some more questions on that topic and we can discuss that. But the pattern is going to be the same. Okay? We can only do the revision through the questions. Why we are doing these MCQ questions? Because it will help you to solve the assignment itself. That's why we are doing these questions. Okay. But till previous year, I was given old year assignment questions which I was discussing. But now, this time, they told me that I should make the questions by myself and discuss with you all. So I am making questions according to my knowledge and whatever is given in the lecture notes. Okay. But still, if you want that, no, this topic was not covered or I missed this topic, then you can let me know which topics you want for the next week if anyone has gone through week 3. If you have not gone, then obviously I will do it my way. But if you want like that, then next Tuesday, come prepared with week 4 or note down the topics which you want in week 4 to be discussed and let me know by week 3. I think numerical questions will come because they are in the lecture notes, so numerical questions will come. I can't say that they won't come. Already a few numericals are there in the lecture notes, right? So you have to go through them or practice them. But I was told not to discuss those in the revision classes. It has to be like how you're getting the assignment questions. That's why I'm avoiding those kind of questions. But if you want me that numerical questions also you want to discuss, then I can keep one or two questions for each session. But it's only that you have to again sit with notebook and pen and you have to solve the answer and tell me. If everyone is okay, then I can have uh, maximum two questions in each week, right? Like I have been told only multiple choice questions, but if you want numericals for practice, we can have two numericals to practice. So from next class, sit with pen and paper. I will show you the question. You have to only tell me the answer. I will only rectify it if you have done it wrong. Okay? Let's say we miss an assignment, what would happen? Nothing will happen, only your attendance is gone. And I don't know what they are going to do with your attendance. Maybe they are keeping it for giving some marks. I don't know. But if you are missing an assignment, obviously the live interaction you are missing. Plus, but you won't miss any kind of source because I am recording the session and also I will be giving you all the PPT. So you can go through the questions again. Okay. So if you are missing an assignment, acha, if you are asking me about your marks based assignment, is it that the assignments you're getting for each week, are you asking about that? If you are missing that, I think your marks will be deducted because each assignment carries marks, right? You're not going to have another assignment test. Whatever you're giving for each week, that is only your test. That's why we're discussing these questions. That I don't know, Ranjani. I'm not sure about. Okay, let's see what is the answer to this question. It is B. Borosilicate glass. Glass is used at lab scale due to fragility. Industrial scale reactors need robust and corrosion resistant materials. So, which material is not typically used? So, the answer is glass. B. Borosilicate glass because of its fragile nature, right? Here you can see the characteristics of material of construction. So what it should be: flexible and durable, non-toxic to reactants and products, resistance to chemicals and metabolic products by the organism, resistance to weathering, low cost and available, easy of fabricate and corrosion proof, high transparency. Okay, so these are the characteristics for the material used in construction of a reactor. More are there. Construction materials differ from small scale, pilot, and large scale glass or stainless steel for small scale processes, stainless steel, mild steel, wood, plastic, or concrete for pilot and large scale processes. So, as here the question was for large scale bioreactor, glass is not applicable. Steel, steel, or concrete is fine as written in the PPT slide. Okay. Moving on to the next question. The draft tube in an airlift reactor primarily functions to control feed rate, increase surface area, improve circulation and oxygen transfer, hold catalyst pellets in place. Start answering seventh question. Tomorrow is the last day for submit week 1 assignment. Try to complete today. Well, I recently joined the course and I'm a bit lagging, that's all. Yeah, so Phoenix, I don't know if that's your name, but as everyone is saying that week 1 deadline is not gone yet, so you can just answer the questions today and submit it. Then I think you won't be having any deduction of marks. Please just check the last date because after the last date is gone for each week assignment, you won't be getting the access to give the answers. Okay, week 2 is also tomorrow, so your friends are helping you, Phoenix. So you can just give week 1 and week 2 by today. Okay. Everyone just go through the industrial biotechnology page. Much information is there which you can go through and know. I think there is some information given on your exam pattern, marks, passing marks. What are the passing marks? Someone was asking last time to get a certificate. So I think it is 40, 40 marks you need to get a certificate. Okay. So yeah, those rules are written already. If I can find out, you can also find out. Okay. So yeah, that's all. Next week, just let me know if you all are able to access to my YouTube videos and the PPT. If you are unable to access, then I have to check once. Okay. Okay, I can't see answers from everyone joined in this session. I think a Google Sheet link was given to you all few days back, last week. Okay, so go and open that Google Sheet. There you can see the Google Drive link for the PPT for week 1 as well as the YouTube link for week 1 video. Okay, try to see. You have got the mail because I have also joined as a learner for the subject and I got the mail. That means you all also got the mail, right? Okay, so let's see what is the answer to this question. It is C. Improve circulation and oxygen transfer. tube creates a circulation loop that enhances gas liquid contact and homogenization. So, the question was the draft tube in an airlift reactor primarily functions to improve circulation and oxygen transfer. Already we discussed what is an airlift reactor. So this is what it looks like. Here you can see the draft tubes in the airlift reactors, which helps in circulation and oxygen transfer. So this, okay. Moving on to the next question. Eighth, a trickle bed reactor differs from a packed bed reactor by using smaller catalyst particles having gas and liquid reactants trickle simultaneously, operating under anaerobic conditions, not requiring any catalyst support. Start answering. So I want to know each of your scores at the end of the session. So please keep noting down how many correct answers and how many wrong answers you're giving out of 15. Okay. Okay. I think we all are done with this question. Let's see what is the correct answer. It is B. Having gas and liquid reactants trickle simultaneously. So that's what is the function of a trickle bed reactor. So trickle bed reactors involve co-current or current counter current flow of gas and liquid over catalyst pellets. So you can see here what is a trickle bed reactor. A packed bed where a liquid reactant is fed from the top, wetting catalytic pellets, and a gas reactant fed either from the top or the bottom flows through the void spaces between the pellets. The gaseous reactant must be absorbed and transported across the liquid film to the catalytic sites at the surface of the pellets. An application is wastewater treatment. So yeah, we are discussing about different types of reactors. So you need to remember what are the function of these type of reactors, how they are different from each other, and what are the application of these kind of reactors. Okay. So this is how a trickle bed reactor looks like. So this is the packed bed and air or gas inlet is here, and this is from where the liquid out is done. Moving on to the next question, which reactor type provides the most uniform temperature and concentration profile? Tubular reactor, batch reactor, CSTR, trickle bed reactor. Start answering. Okay, is everyone done? Let's see what is the correct answer. It is C, CSTR. So, which reactor type provides the most uniform temperature and concentration profile? It is the CSTR. Due to vigorous stirring, CSTRs maintain homogeneity in temperature and concentration, right? So here you can see we already discussed about this figure, continuous stirred tank reactor. So here, because of this continuous agitation, there is always maintenance of temperature and concentration in the reactor. Moving to the next question, the degree of reduction of a compound indicates its molecular weight, its pH level, number of electrons available for transfer, amount of oxygen required for oxidation. Start answering. Okay, let's see what is the correct answer. It is C, number of electrons available for transfer. So the degree of reduction of a compound indicates about the number of electrons available for transfer. It quantifies the reducing power of a compound important in balancing biochemical redox reactions. So here you can see the slide. It is about oxygen demand. The requirement for oxygen is related directly to the electrons available for transfer to the oxygen. The electron balance for oxygen demand, numbers of available electrons in the substrate plus the number of available electrons in O2 minus number of available electrons in the biomass plus the number of available electrons in the product. So here we are talking about this thing, right? Which indicates the number of electrons available for transfer, right? Moving on to the next question. In a fed batch reactor, no product is formed. Products are removed continuously. Feed is added during operation but not removed. Reactor operates under steady rate. Oh, sorry. Already the answer is given. I forgot to remove it. So yeah, just we'll discuss this. Answer is C. Feed is added during operation but not removed. This is what is in the fed batch reactor. Limited substrate is added during operation. Product is harvested at the end. Here you can see from the slide, Fed batch reactor, a tank in which reactants are charged initially and limiting reactant is added continuously during the operation. So the limiting reactant is added continuously. That is why you can see that it is being fed continuously. No product removal during the process. Applications are bakers, yeast fermentation process, pharmaceutical industries like secondary metabolites. So here you can see batch reactor, fed batch reactor and continuous reactor. What are the differences? You can see here the symbols are given, right? Moving on to the next question. In which reactor type does plug flow behavior help minimize product inhibition? CSTR, fed batch reactor, tubular reactor, packed bed reactor. Okay, I will show you the previous question again after all the sessions are done. Question number 10. Okay, I will show you question number 10. For now, answer the questions. After all the questions are done, I'll go back to question number 10 once again. So this is question number 12. Start answering it. Okay, so it is asking for access. Okay, I will let that access go open for everyone. I'll do it after the session is completed. Okay, let's see what is the correct answer to this question. It is C. In plug flow, fresh substrate is always at the inlet, limiting product accumulation and inhibition. So that is about the plug flow reactor. We have already discussed this before. So we'll move on to the next question. The main difference between a bubble column and an airlift reactor is presence of agitation in bubble column, use of external air supply in airlift, use of a draft tube in airlift reactor, gas is introduced at the top in bubble column. Start answering. Okay, let's see what is the correct answer. It is C. Use of a draft tube in an airlift reactor. So, as we already discussed, airlift reactors have draft tubes, whereas bubble columns do not have. So, the draft tubes are used to improve circulation in the airlift reactors, right? So, as we have already seen, there are draft tubes in the airlift reactors, whereas the bubble column reactor does not have any kind of draft tubes, right? Moving on to the next question, a material commonly used in photobioreactors for transparency is mild steel, epoxy coated steel, stainless steel, borosilicate glass. Start answering. Okay, let's see what is the correct answer to this question. It is D. Borosilicate glass is used where transparency is needed. Example for light exposure in photobioreactors. Okay. The borosilicate glass is the only option out of the all which you can think by common sense also that it has transparency which can be used for photobioreactors for light exposure. We have already discussed this slide as well. Moving on to the last question of today, which of the following is not a characteristic requirement for reactor construction material? High cost, non-toxicity, corrosion resistance, durability. Okay, this one also I have kept the answer. Okay, so the answer is A, high cost, right? Which of the following is not a characteristic it was written? So that's why the answer is high cost. Low cost is a desirable trait, high cost is not favorable for scale up and industry, right? We have already discussed about the characteristics for construction of a reactor, right? So, yeah, this was it for today's session. So, someone was asking question number 10. So I'll quickly go back to that question. So yeah, this is question number 10. The degree of reduction of a compound indicates number of electrons available for transfer. So it quantifies the reducing power of a compound important in balancing biochemical redox reactions. So this question is taken from this slide. Okay, is it clear now? Yeah, glass is transparent. That's why it is used for photobioreactors because light exposure is needed, right? 14th question answer, ma'am. Okay, 14th question is a material commonly used in photobioreactors for transparency is D, borosilicate glass, right? Is used when transparency is needed. Example for light exposure in photobioreactors. Is there any confusion in this question? Okay. So we are done. I think two answers I have only shown you, so you can deduct that. So out of total 13 questions, what is your score? Let me know. 10 out of 13, 12, 11. Okay. Is there no one? 12 is a very good score, Shreya. Is there no one who got 13 out of 13? And you people need to practice more because every question was from the slides, right? Just go through the slides a little bit more in detail, and I think you will get full marks as well. Okay. Yeah. Is there any doubt? I will give access. I will check why you are not getting the access, and I will do it. Other than that, is there anything you want to say? Then you can say, or else we will disperse. My God, there's a big message. While joining the course, it was written that this course is helpful for job selection and top biotech countries like so certificate is enough. Actually, and many say there is very less chances of placement of smart college and students rather than students. Okay, Abba hai. So first of all, I'm not from biotechnology background, and I'm really not sure about this. Why students are so students are either to do those salary jobs, and I'm also not sure what was told to you to get to this course. So, but I am 100% sure that this certificate will help you if you are really, means it will add up to your CV, obviously, if you are trying to go for some biotech industries. But for going to any kind of job, I think your CV needs good marks and good experience. Generally, they see good experience, or so this is also counted as an experience, right? You took a NPTEL course, so these are really good certified courses. Um, but yeah, this thing I have also, for not just for biotechnology, for all the people in biology, they have heard this that there is no scope, you have to struggle a lot. I have also heard it, but that is not true. As you go with the flow and you keep joining somewhere or do whatever you want, I would suggest don't take a gap anywhere, and yeah, I think you will land up well. Ma'am, can we use the certificate for internship? Okay, so everyone, I am not sure about any of this. I have no information. There is no information given to me about any of the details of your classes. For that, you have to mail to the course instructor how the certificate will be helpful, or you have to mail to the NPTEL team. They can only tell you. So I belong to so currently I'm doing my PhD in animal biology department, but my research focuses on molecular biology and protein biology, you can say. So and my MSc is in molecular medicine, and my BSc is in zoology. So yeah, you can say animal biology background, and but I was chosen for this course, so I had to go through the lectures to make sure that I am capable enough to teach, to discuss with you all the questions. I'm not sure how much good I'm doing, but yeah. So, okay, is there anything else? I don't know how much I was able to satisfy your question, Abhay. How can this course be helpful for botany background students? I don't think this is anyway related to botany at all. So if you are from botany background, I'm not sure of. Only it will be a kind of experience that you added to your CV. That's all. You got to learn something new. But if you want to pursue a botany career, I'm not sure about that. Yeah, microbiology, obviously, it is somewhere related to microbiology. In microbiology, also, we go through these reactor topics and everything. So, yeah, from whichever biology background you are, if you are taking an NPTEL course and you're getting a certificate for it, obviously it will be helpful for you in the long run. Obviously, you got something to add to your CV, then obviously it is helpful, right? If botany, you can take plant biotechnology course as well. Yeah. If plant biotechnology is there, you could have taken plant biotechnology, right? But you all have joined in this course called industrial biotechnology with some sense, right? You must have thought about some reason for which you opted for this course, or you were given no choice, but you had to opt for this course. I'm not sure about that. You have your own reasons, but I can just say that if you are learning a new topic, a new lesson which you are not related to, that is also helpful. And if you are learning something which you already know and you are acquainted with, that is also helpful. Anyhow, you are going to get a NPTEL certified, like Swayam MOOC certified certificate. So these courses are designed by the government itself, right? So that is always helpful, right? Even when I was in my masters, I had to take an NPTEL course, um, and I also took it. Okay, okay, everyone. So we'll meet next week, uh, Tuesday, 7 o'clock. Everyone, please be on time and try to come prepared. I will try. I will see what are the numericals kept for week 3. If it is possible to. Okay. Thank you, everyone. If